May I start, sir? A very good afternoon, respected director, sir, faculty members, dear student and audience. I, Dr. Sant Kumar Verma, Associate Professor, ISF College of Pharmacy, Moga, would like to welcome Professor Ranju Bansal, Ma'am, Dean, Faculty of Pharmaceutical Sciences and Professor, University Institute of Pharmaceutical Sciences, Panjab University, Chandigarh, at the online platform of ISF College of Pharmacy, Moga, for delivering talk on Xanthine, a privileged scaffold. I request to respected director, sir, please welcome our today's speaker, Professor Ranju Bansal, ma'am. Sir, please welcome, ma'am.
So, thank you very much, Dr. Sant Kumar Verma ji. Good afternoon, everyone. On behalf of ISF College of Pharmacy Family and ISFCP Dialogue Series, I welcome Dr. Ranju Bansal from core of my heart. As you know that this series and purpose behind of this series to provide the technical, scientific, purified knowledge to the audience, researcher, a student, faculty member, so that we can connect to each another. As we know that pharmacy profession is very small and number of the dynamic, dedicated, young researcher working in the different field like a Dr. Ranju Bansal. So this is the opportunity to each and everybody to connect the peoples in a easy way through the YouTube mod of the ISF College of Pharmacy. So again, I welcome Dr. Ranju Bansalji for accepting our invitation and provide your very precious time. And I hope that your talk and today's discussion it will be the very helpful to the all the researcher and pharmacy professions so that person they can be follow and take the advantage of this types of the talk. So thank you very much. Without consuming time, I request to Dr. Sant Kumar Barmaji, please proceed.
Thanks, respected director, sir. Now I would like to invite Professor Ranju Bansal, ma'am, for her talk. Over to ma'am.
Okay. Okay, so at the offset, I would like to thank Professor Gupta and Dr. Sant for giving me this opportunity to interact on this online platform. So, the topic which I have chosen today is a very general and basic topic, that is xanthine, a privileged scaffold. So, privileged means, in the medicinal chemistry point of view, we say privileged means versatile. So, there are some scaffolds or chemical moieties or the chemical frameworks which are extensively used for drug discovery and development. And for example, xanthine is one of that privileged scaffold. Others are dihydropyridines, benzodiazepines, steroids, etc. So I have chosen here xanthine because in one or the other way we come across xanthine in our day-to-day life as well. So xanthine is a
Ma'am,
purine
uh,
base.
ma'am, excuse me, ma'am, please
Am I
share.
audible?
Yes, ma'am. Ma'am, please share your slide.
I'm already sharing.
Ma'am, it has been stopped. Please share again.
Are these visible?
Yes, ma'am.
So should I start again?
Yes, ma'am. Yes, ma'am.
Okay. So I'm starting from the beginning now. Okay, so I would like to thank Professor G.D. Gupta and Dr. Sant Kumar for providing me this opportunity to interact with all of you on this online platform. And the topic which I have chosen is a very general topic, that is xanthine, a privileged scaffold. So privileged in from the medicinal chemistry point of view, we consider certain chemical moieties or the frameworks which are widely explored for drug discovery and development. So xanthine is one of those moieties, and the others which can come under this category are dihydropyridines, steroids, benzodiazepines, etc. So, xanthine is a purine base found in most human body tissues and fluids and other organisms. A number of stimulants are derived from xanthine. For example, caffeine, theobromine, and theophylline. So, if you look at the structure of xanthine, so we have three nitrogen with one hydrogen attached to them. So, if any of these hydrogens they are replaced by methyl groups, then it can lead to the development of caffeine, theobromine, and theophylline or paraxanthine. So, one by one, we'll be studying these xanthines. So, the natural sources of these methyl xanthines they are caffeine, theobromine, and theophylline, which are produced. And so, caffeine, theobromine, and theophylline they together constitute methyl xanthines. And caffeine is obtained from tea leaves, coffee beans, cola nuts, cocoa seeds, and similarly, theobromine and theophylline, they are also obtained from tea leaves. However, theobromine is present in cocoa seeds and theophylline is present in criollo beans. So, caffeine is a bitter white crystalline xanthine alkaloid and is a stimulant drug. So, we all know that in our day to day life, we are not, unless and until we take one cup of tea, so we are not stimulated enough to do our day to day activities. And in the morning itself, one starts the day with the cup of tea. So, this is the world's most widely consumed psychoactive drug. But unlike many other psychoactive substances, it is legal and unregulated in nearly all parts of the world. So nobody can stop you from taking tea. So this is legal and is also unregulated. So beverages containing caffeine such as coffee, tea, soft drinks, energy drinks, they enjoy great popularity. In North America, 90% of adults consume caffeine daily. So caffeine is one of the methyl xanthine. So this is an achiral molecule without any stereoisomers. So if you can look at the structure, then there are no chiral carbons present. And the two amide groups of caffeine exist predominantly as Zwitter ionic resonance structures, where the nitrogen and carbon atoms are double bonded to each other, so that both of these nitrogen atoms are essentially planar in sp2 orbital hybridization. So this fused ring system contains a total of 10 pi electrons and according to Huckel's rule is aromatic in nature. So looking at the health effects of caffeine, so it can have positive as well as negative effects. The positive effects include increased attention and alertness and decreased fatigue. So you can correlate it with when you're very tired, you take a cup of tea and you feel very attentive and alert and your fatigue goes. And it also, but the quantity of caffeine or theophylline or theobromine is so less in that it doesn't affect the other functions of the body. So lower risk of cardiovascular disease is another advantage, and lower risk of diabetes, increased metabolic rate. But there are some negative effects if you take a lot of caffeine. So one can be anxiety and then addiction. So I think the tea addiction, most of the people are we suffer, and we tend to have headaches if we don't take tea regularly. Then increased vasoconstriction and blood pressure is another negative effect. Reduced control of fine motor movements and stimulation of urination. So these can be some of the negative effects, but mostly these occur if it is taken in high quantity. So second methyl xanthine is theobromine, and it is derived from the name Theo meaning God and Broma meaning food. So it is food of God. So it has a faintly bitter taste and it has slightly solubility in water. So the difference in theobromine and caffeine is that one of the nitrogen is not methylated here. So then this is also present in cocoa beans up to 1.5 to 2.5%, and generally used as a diuretic in form of its double compound with sodium salicylate, although we have now better diuretics available. And it is also a cardiac stimulant and remedy for dropsy. So dropsy is edema type which occurs in the body. So because it is diuretic in nature, so can be used for that purpose. Then therapeutic uses it can act as vasodilator, diuretic, heart stimulant, and treatment for other circulatory problems, including arteriosclerosis, certain vascular diseases, angina pectoris, hypertension, and also for treatment of edema, as I just told you. So then the third one is theophylline, and this is 1,3-dimethylxanthine. So the nitrogen at 7 position is not methylated here, and this is mainly used for respiratory diseases such as COPD and asthma under a variety of brand names. So as a member of xanthine family, it bears structural and pharmacological similarity to caffeine. So if we go for the numbering of this nucleus, then one can start from the nitrogen, which is having the methyl, and then it can go 1, 2, 3, 4, 5, 6, 7, 8, 9. So 1,3-dimethylxanthine is theophylline, 1,3,7-trimethylxanthine is caffeine, and 3,7-dimethylxanthine is theobromine. So this again is present in cocoa beans, and amount as high as 3.7 milligram per gram have also been obtained from Criollo cocoa beans. And trace amounts are present in brewed tea, and although it is quite less, that is 1 milligram per liter, which is significantly less than the therapeutic dose. The therapeutic uses of this theophylline, they are relaxing bronchial smooth muscles. That's why it can be used for the asthma or respiratory problems. It can also increase heart muscle contractility and efficiency, and increases heart rate, increase blood pressure, renal blood flow, and it also has anti-inflammatory effects and also CNS stimulatory effects. And it mainly produces the CNS stimulatory effects by acting on the medullary respiratory center. Now, these were the main methylxanthines, theophylline, theobromine, and caffeine. Now, these like in natural sources, these are present in very small amount and they do not produce very high effects, but consuming these in day-to-day life can produce some of the effects. But if we can alter the structure of this privileged scaffold, that is xanthine, as I've told you that this is a versatile nucleus and it has got many centers which can be structurally explored to produce new drugs, and those drugs can act on they can be used for various purposes. So the mechanism of action of xanthines is through various modes, and one of the mode is by targeting the adenosine receptors. And adenosine receptors are widely present in the body, and they occur throughout the nervous system and also in vascular endothelium, heart, liver, adipose tissue, and muscles. So the actions that result from xanthines are dependent on which type of adenosine receptors it blocks, in which tissue the receptors are located. So most widely used psychoactive drug in small, so this xanthines, they remain the most widely used psychoactive drug. The small amounts, they no long-term effects have been noted. Even small amounts taken regularly can produce withdrawal effects. As I earlier said that not taking tea for the people who regularly take tea can lead to headaches. So even the small amounts can lead to the effects. But the large amounts, they can also produce adverse symptoms such as anxiety and insomnia. However, we can modulate the structure of xanthine in such a way to produce the beneficial effects. So one was on the adenosine receptors. So how methylxanthines, they can be used in CNS. So they are widely used to promote wakefulness and counteract fatigue and induced by restriction of sleep. And so I would like to point out, whenever you have to remain awake, when you want to remain awake throughout the night, then you have to take 2-3 cups of tea or coffee. So xanthines, they bind to the adenosine receptors, and the antagonism of the adenosinergic system is believed to be the mechanism through which xanthine counteracts sleep in humans as well as in other species. So again, they increase alertness, wakefulness, increase in faster, clearer thinking, increased mood, sense of well-being, decreased fatigue, decreased sleep, and increased skeletal muscle activity. The central effects of psychostimulants other than caffeine, so it is one of those. So amphetamine, cocaine, caffeine, they can increase the motor activity. They can also produce arousal, reinforcing effects, and subjective stimuli. So the other advantage of methylxanthines is in Parkinson's disease. So we know that and it is said that person who regularly take tea throughout their life, 1 or 2 cups of tea, it is very it is said that these people, they might not suffer from the Parkinson's disease. So because slowly this theophylline or caffeine is going inside the body and producing the desired effects. So Parkinson's disease, we know, is a chronic progressive neurodegenerative disorders, and it involves degeneration of nigrostriatal dopaminergic system, leading to the striatal dopamine depletion. So this depletion of dopamine can lead to the inactivity of the skeletal muscles. And people have established an association between the consumption of coffee or other caffeinated beverages with a reduced risk of developing Parkinson's disease. And this effect of methylxanthines is thought to be linked with the blockade of adenosine A2A receptor subtype by caffeine, as the adenosine A2A receptor controls locomotor behavior and neurotransmitter release in basal ganglia. So otherwise, for Parkinson's disease, one will go for the levodopa, which is a dopaminergic therapy, which will fulfill the requirement of dopamine. But here, this is a non-dopaminergic approach for the treatment of Parkinson's disease. So there are some drugs which are already approved for the Parkinson's disease, which are based on the xanthine scaffold, and some of these leads are KF17837, KW6002, and istradifylline, which is already approved by FDA for the Parkinson's disease. So A2A and D2 receptors, they have opposing effects on adenylate cyclase and CAMP production in cells, such that activation of A2A receptors inhibits dopamine D2 receptor signaling. So if the xanthines, which act as A2A receptor antagonists, they enhance the D2 dependent signaling, so can be used for the Parkinson's disease. So then methylxanthines, they can also be structurally explored for the treatment of seizures. And theophylline can trigger seizures in patients without known underlying epilepsy and is an added risk factor. So because xanthines can produce such seizures, so structural exploration might produce some compounds which can be used for this treatment. So caffeine can inhibit the binding of benzodiazepines to GABA receptor, which might contribute to a convulsant role of high or toxic doses of methylxanthines. So as I've already told you, methylxanthines can act as diuretics. So they increase urine output until more potent diuretics became available in the middle of the last century. So the diuretic potency of methylxanthines is found to be theophylline is the most potent diuretic in comparison to caffeine, paraxanthine, and theobromine. Methylxanthines, they are weak renal vasodilators and act as competitive antagonists against adenosine-induced vasoconstriction. Caffeine and theophylline, they stimulate the secretion of renin by inhibition of adenosine receptors and removal of the general inhibitory brake function of endogenous adenosine. So then methylxanthines, they also have cardiovascular effects. So the cardiovascular effects are due to the antagonism of adenosine A1 and A2 receptors and inhibition of phosphodiesterases or mobilization of intracellular calcium. That requires much higher concentration, but these are also two factors which can be explored using methylxanthines for inhibition of PDE enzymes and the mobilization of intracellular calcium um, for the CVS effects. Methylxanthine derivatives, they are used in patients for their distinct cardiovascular effects, such as pentoxifylline in patients with peripheral artery disease. At high concentration, methylxanthines, they can inhibit phosphodiesterases and resulting in augmentation of the beta adrenergic rise in intracellular CMP levels. So one very important use of methylxanthines is in asthma, and still in till now we are using theophylline derivatives for the treatment of asthma, and the most popular compound displaying non-specific PDE4 inhibitory activity is theophylline, and this is prescribed worldwide for treatment of asthma, and it is suggested as an additional bronchodilator when moderate to high doses of inhaled steroids fail to control asthma symptoms. Even during COVID times, salbutamol or methylxanthines were taken for the relief from the respiratory disorders. So xanthines, they cause bronchodilation by relaxing smooth muscles, and relief of bronchospasm and greater airflow in and out of the lungs takes place. And the one representative example is aminophylline. So these are the uses of xanthines. Then how, through which different molecular mechanisms they act. So as I've just told you, they can also act as calcium releasers, can also produce phosphodiesterase inhibition, then GABA A receptor inhibition, and antagonism of adenosine receptors. So in these two areas, phosphodiesterase inhibition and antagonism of adenosine receptors, xanthines have been widely explored, and otherwise, for producing these effects in general inside the body, high doses are required. But for the antagonism of adenosine receptors, xanthines have been widely explored. So for producing the calcium release from intracellular storage, you require at least 250 millimolar, and maximal effect can be produced at 5 to 20 millimolar. But this mechanism is unlikely to account for the effects of caffeine in humans because of its low potency. So you require as high as 250 millimolar to produce the calcium release. Then phosphodiesterase inhibition. So they also inhibit this enzyme, and this enzyme is responsible for the degradation of cyclic AMP to cyclic GMP, and it is proposed that caffeine operates by elevating intracellular CMP concentrations. Because xanthines or caffeine will inhibit phosphodiesterase enzyme, so cyclic AMP will not be degraded and its levels will be increased. But this mechanism is unlikely to account for the effects of caffeine in humans in day-to-day life I'm talking about, because PDE inhibition requires caffeine concentration 10 to 100 times higher than those achieved by typical dietary intake. Then GABA inhibition also requires Ki of 280 millimolar, and it is suggested that normal human caffeine consumption is unlikely to produce caffeine doses high enough to inhibit GABA A receptors. So then we come to the adenosine receptor antagonist, which is the mechanism which has been extensively explored for xanthines for producing various desired effects. So these are non-selective adenosine receptor antagonists, and the natural plant alkaloid caffeine and theophylline. They were the first adenosine receptor antagonists described in the literature. And another simple natural xanthine, theobromine, was found to have only weak activity as adenosine receptor antagonist. So they exhibit micromolar affinities and are non-selective. So we need to understand a little bit about the adenosine and adenosine receptors. Adenosine is a purine nucleoside. So you can see that it has lot of structural similarity with the xanthine, and it consists of a base adenine which is attached to D-ribofuranose through beta glycosidic linkage, and this is structurally and metabolically related to nucleotides such as adenosine monophosphate and adenosine diphosphate and adenosine triphosphate. So adenosine produces its biological effects and is continuously formed intracellularly as well as extracellularly. The free cytosolic concentration of adenosine is reported to be 0.8 millimole per liter. So adenosine inside the body keeps on acting on various adenosine receptors to produce the desired effects. And looking at the various adenosine receptors, so these are mainly categorized into four classes: A1, A2A, A2B, and A3. And the adenosine receptor has the extracellular domains of the N terminus and three extracellular loops, while the intracellular domains of the C terminus and three intracellular loops. So on these adenosine receptors, xanthines, they produce their effect. So as of now, if we talk about the natural products like caffeine and theophylline, so they act on adenosine receptors, but their effects are non-selective. So if we want to produce some selective inhibition of the adenosine receptor, then we can structurally explore this privileged scaffold to get high potency and more selectivity for a particular receptor to produce the desired effects. So this is our signaling pathway. So I'll just quickly go just past this. And this is a more important slide, that how the adenosine receptors, they are distributed inside the body and what type of actions they mainly produce. So A1 adenosine receptors, they are mainly present in the CVS and adrenal gland, then brain, skeletal muscles, liver, kidney, etc. But majority of the functions of A1 adenosine receptors, they are related to the cardiac protection action, and then diabetes, asthma, nociception, renal failure. Then A2A adenosine receptors, which have been widely explored for the treatment of Parkinson's disease. So this receptor is important from this point of view, and the antagonism of this receptor can find out a remedy for the Parkinson's disease. Then A2B receptors have been vastly explored for the treatment of asthma and respiratory disorders. And so you can have a look at the distribution of these A2B receptors inside the body. Then A3 receptors, they are mainly present in the cerebellum, then hippocampus, thyroid, etc. And they are mainly involved for neuroprotection. So till date, most of the xanthines or the xanthine privilege structure has been explored for A2A and A2B receptors. So these three I've already discussed. These are the three xanthines which are the naturally occurring ones, and they are non-selective antagonists of adenosine receptors. So then these are the various structures which have been like the basic nucleus has been explored to produce the better agents. And this evaluation of a series of monosubstituted xanthines showed that substitution at N1 was most important for all AR subtypes. So it is said from this preliminary study that if this nitrogen is substituted, then it can bring a lot of changes in the activities for the adenosine receptors. Then this is the various like observations which have been made from the medicinal chemistry point of view, that a 1 propyl residue at N1 is best for A2B and A3, and 1 benzyl residue was optimal for A1 and A2A. So on, 1 propyl xanthine, that is the compound 4, where R1 is propyl. So it shows high affinity of small simple xanthine derivative for human A to B. So you can see that from the non-selective and low potency of millimolar, Ki has been reduced to 360 nanomolars with only one substitution of propyl group at N position. And selectivity was also observed at least 7-fold through other subtypes. So this is why this is called as a privileged structure, that changing one methyl to propyl increase the potency towards A to B so to many folds, and also increase the selectivity. So in amprophylline 6, that is 3 propyl xanthine. So this is the 6th one, where R2 is the propyl group. Others are these H. So this is less potent. Ki is 4730, and but more selective, at least 14-fold. So this is how the versatile pharmacophores they behave. Small changes in their structures bring drastic changes in the biological activities. So another type of modification, that is elongation of 1,3-dimethyl group. So if you elongate the alkyl groups present at N1 and N3 from to propyl or to larger, so substitution with 1,3-dipropyl led to the formation of compound 8 with increased affinity at RAT A1AR by approximately 20-fold. So if only propyl is present at this position, then it is more selective for A to B. If you shift the propyl to the other position as well, then increases the activity for the A1 adenosine receptor. Similarly, presence of a propargyl group at one position. So this was the first A2 adenosine receptor selective antagonist described in the literature, 3,7-dimethyl and 1 propargyl. But the degree of selectivity versus A1 adenosine receptor was low. And if you compare it with 7 unmethylated, so no methylated group is present here, then it indicated that 7-methyl group lead to large decrease in A1 adenosine receptor affinity, but increase selectivity to A2A or A2B. So coming to some more 8 substituted xanthines. So as I've told you, these 1, 3 and 7 nitrogens, they have been widely explored for increasing the potency as well as selectivity towards adenosine receptors. Then 8 position of the xanthine skeleton has been widely explored, and introduction of 8 aryl substituent, it increases the A1 adenosine receptor affinity. But later on it was found that the exploration of this benzene at 8 position, it led to the development of A to B adenosine receptor antagonists, and many of the agents, they have A to B adenosine receptor antagonistic activity in nanomolar range. So 1,3-diethyl 8-phenylxanthine displayed Ki value of 44 nanomolars at the RAT A1 adenosine receptor, and also shows selectivity. And homologation of 1,3-dipropyl group in the 8-aryl analog provided a desired boost in affinity. However, the unintended consequence of rapidly diminishing aqueous solubility made this series unable to be used in typical pharmacological studies. So as a medicinal chemist, one has to take care that your structural exploration is not only producing the compounds which are highly potent, but they should also be having drug like properties. So from 1,3-propyl, when the alkyl groups were extended with the desire to produce compounds which are highly potent, then it led to decreased solubility. So from 8 substituted coming to 7 substituted along with the 8 substitution. So this led to the first highly potent and selective A2A adenosine receptor antagonist. And it has been widely observed that substitution of a methyl group at N7 position leads to A2A selectivity. So you can have the molecules which can be used for the treatment of Parkinson's disease. So these compounds here, you can see that all the different positions of the xanthine skeleton, they have been widely explored. This compound, this is the, so here you can see that at N7 position, there's no methyl group, but modifying the structure to producing a cyanoderivative. So this produces a highly selective adenosine A2B adenosine receptor antagonist with nanomolar affinity at human receptor. And this compound is also used in radioligand binding assays as a radiolabeled substance for or as a standard. So further substitution of uncharged electron donating groups like a methoxy group or a hydroxyl group at the para position of the 8-phenyl group also enhances the A2B adenosine receptor affinity. So then coming to the work which we have carried out in our laboratory, and we not only explored this xanthine scaffold for the A2B selectivity, but also for A2A selectivity. So we have explored this moiety not only for the treatment of asthma, but also for the treatment of Parkinson's disease. So you can see that this privileged scaffold, so you can increase or decrease the chain lengths at different positions, N1, N3, N7, and then various substituted groups you can put at 8 position to produce the desired xanthine derivatives. So I have not gone much in details of the exploration which we have carried out in our laboratory for producing the compounds, but I'm just trying to show that all these positions can be explored to produce compounds or desired compounds with desired potency and selectivity for a particular adenosine receptor. So we also explored the effect of various alkyl groups at 1, 3, and 7 position, and it was observed that most dramatic alterations, they are produced on substitution at the 8 position, and synthesis and pharmacological activity of a variety of xanthine derivatives were studied. So I will just like to go to the one synthetic methodology, that is general scheme for the synthesis of 8 substituted xanthine derivatives. And to start with, it seems very simple technique, synthesis of 5,6-diamino uracil, which is the starting material used for the synthesis of 8 substituted derivatives. But to tell you that the synthesis of this diamino uracil almost took us one to 2 years to get this compound, because this is highly unstable and it requires particular conditions to form this compound, which is a starting material for preparing the 8 substituted derivatives. So this is a key intermediate to the synthesis of all 8 substituted xanthine derivatives. And this is the general method for formation of this. So you can condense this dimethylurea with cyanoacetic acid in presence of acetic anhydride to produce 6-amino-1,3 dimethyl Then this is converted to nitroso derivative in the presence of sodium nitrite and acetic acid, and then in presence of sodium sulfite converted into diamino uracil, which is, again I will reiterate, quite difficult to get, unstable in nature. Once you get this, nobody can stop you from making the 8 substituted xanthine derivatives. So coming to the conclusions, xanthine derivatives are the most potent adenosine receptor antagonists reported to date. The purine skeleton possessing vulnerable 7 and 8 positions has given an excellent opportunity to study the structure-activity relationship within the series of xanthine derivatives, which could be explored using rational drug designing approach. It is expected that synthetic modifications of these naturally occurring molecules might lead to selective and medicinally active chemical moieties with minimal side effects for the treatment of various pharmacological disorders. In all, it has been observed that substitution pattern and type of substituent not only at 8-phenyl group, at other positions as well, greatly affects the affinity and selectivity of xanthine derivatives at adenosine receptors. It can be concluded that suitable selection and positioning of aryl substituents may lead to development of potent and selective xanthine-based adenosine receptor antagonists. So, thank you so much. And once again, I would like to thank Professor Gupta for providing me this opportunity for delivering on this online platform. Thank you so much.
Uh, Thanks, respected madam, for excellent talk on xanthine and exploring entire medicinal chemistry of xanthine in relation to adenosine receptor. I would like to invite director sir to thank madam.
So, thank you very
Just a
much,
second.
madam. Very nice lawyer talk, very smooth, effective, and you have covered all the aspects which is related to the Xanthine. And obviously, this is the very very important because most of the persons, as you have initial slide, the consuming the caffeine, and for this purpose, we no need the any permission and no need the you can say any types of the things related to government, family, or the society like that. But obviously, if we need the more pharmacological, effectively useful to this one, for this purpose, need to the further work as you have mentioned. If it can be modified in the more and more and form, then it will be the more and effective and useful. So thank you very much, madam.
Thank you.
And I hope that your talk it will be the very very useful to each and everyone, those who are related to and working on this topic. So thank you very much.
Thank you so much. Thank you.
Thank you, Dr.
Thanks,
Sun,
sir.
for
Thanks,
organizing this
ma'am,
lecture.
for sparing. Yes, sir. Thanks, ma'am, for sparing your valuable time for ISFCP 101 dialogue series. I want to request the audience if you have any query related to lecture, please write in the chat box. We will reply after discussion with the expert. Thanks, ISFCP management, director, sir, and ISFCP coordinator IQAC, Dr. Siddharth Mehan, for organizing this dialogue series. Thank you everyone for being with us at ISFCP online platform. Thank you everyone.
Thank you so much. Thank you.
Thank you.
So, should I leave now?
Yes, ma'am. Yes, ma'am.
Okay, okay. Thank you.
Thank you.